Hey, this is Mrs. Dellosso, and welcome to our very first podcast. So, in this video, we're talking about the scientific method. I'm sure you've learned about the scientific method before, so this video will be more of a brief review or recap of the scientific method. There are 3 objectives, though, that you need to know by the end of this video. So I suggest you write them down so you make sure you follow through with them. The first objective would be to list and describe the steps of the scientific method. The second objective would be to explain the differences between the control group and the experimental group. And the third objective is make sure you can explain the difference between the independent variable and the dependent variable. All right, so let's begin. Alright, so how can we define the scientific method? The scientific method is a logical procedure for choosing an answer to a question. And let's go into some of the steps that are involved with the scientific method. Alright, the first step we're going to talk about is defining the problem. For example, a problem could be, do geraniums grow better in dim or bright light? Make sure it's something specific. Something like what makes plants grow is really not specific and can be tough to test. The second thing to do is to do the preliminary research. What can you find out about geraniums and plants in general? What do they need to grow? And also, the next thing you're going to do is to form a hypothesis. So these 3 steps here are basically the steps that you'll do before you actually get into your experiment or survey. Let's not forget what a hypothesis is. A hypothesis is an educated guess that attempts to answer a problem. It is always in the form of a statement and never in the form of a question. And typically, I like to have my students write their hypothesis in an if-then format. And we're going to take a look at an example of a hypothesis later on when we look at an experiment. So after you've developed your problem, done the research, and came up with a hypothesis, here are the following steps. Then you're going to set up your experiment or survey. For example, you might have a question similar to what kind of tree is most common in a certain region or area, and that would be best answered through a survey. So after you set it up, you're going to observe the experiment or survey, collect and record the data from your experiment, and then you're going to classify the data, and you can think of classifying the data as another way of organizing your data so it can easily be read. All right, next step, number 5, you're going to analyze the data, and that means you're going to determine what the data reveals about your problem. Number 6, you're going to choose the answer. In other words, what solution does your data point to? If the data implies a conclusion different from your hypothesis, that's when you're going to revise your hypothesis and do the experiment again. Number 7, you're going to verify the answer, making sure that it's valid. And by valid, we mean accurate and reliable. So repeat the experiment. Do you get the same or similar results every time? So the more times you repeat the experiment and get the same results, the more valid the answer is. And then number 8, you're going to predict future results. The goal of using the scientific method is to be able to draw conclusions that can then be applied to similar cases. All right, so let's take a look at the controlled experiments and what's involved. There are typically 2 identical groups that differ in only one variable. And the one variable that's going to be different is the independent variable, and that's the factor that is being tested. So the experimental group is the one that we're exposing to the independent variable, and the control group is the group that won't be exposed to the independent variable. Now keep in mind, the control group and the experimental group are going to be identical in every aspect except the independent variable. The dependent variable is what's going to be measured in the experiment. So the independent variable is the factor that's being tested or manipulated. Dependent variable is the factor being measured. Now, something that we have to keep in mind, if there are multiple variables in an experiment, that can cause the experiment to be invalid. For example, if you're including more than one independent variable, there's a good chance that you're not going to know which variable was responsible for your observed results. So that's why we always keep it to one independent variable, and also make sure, again, you repeat the experiment and keep your sample size large. All right, so let's take a look at a sample experiment. So let's develop our problem here. Will a newly developed hair growth drug promote hair growth? So here's a potential hypothesis. So let's say we're testing this on mice. So if mice are given the special hair growth drug, then the mice will grow longer fur. There's that using that if-then format, and remember, a hypothesis is in the form of a statement, not a question, and this is something that we can test. So let's take a look at our setup. Notice we have our control group and our experimental group, and notice in our two groups here, everything is exactly the same. There's two sets of mice grown in identical cages. They're in the same room. They're going to have the same diet. The only difference is that the experimental group is going to receive the independent variable. In this particular experiment, the independent variable is going to be the hair growth drug. So now let's take a look. After we run our experiment, we can see there is a difference between the two groups here. So we can see here that there was definitely a change between those two groups. So again, in order to make our experiment more valid, we would repeat the experiment and maybe use a larger sample size as well. All right, make sure you have your organized notes and then take the Check for Understanding quiz, and we'll go over it in class.